So I can't blame anyone here if they're in a little state of shell shock. So we have an expert set of panelists who are supposedly more immune to shell shock because they're so expert. And plus, we did give them a little bit of advance notice. So they haven't just heard these ideas in the last hour. Let me introduce the panelists. Our first speaker is going to be Dr. Steven Spielberg, Deputy Commissioner for Medical Products and Tobacco at the FDA. He's a pediatrician and a pharmacologist, and was most recently the Marion Merrill Dow Chair in Pediatric Pharmacogenomics and Director of a Center for Personalized Medicine and Therapeutic Innovation at Children's Mercy Hospital in Kansas City. Our second distinguished panelist is Stephen Evans. He's a professor of Pharmacoepidemiology at the London School of Hygiene and Tropical Medicine, immediate past president of the International Society of Pharmacoepidemiology. After training in physics and chemistry, he worked in statistics and computing at the London Hospital and Medical College for 25 years, then the head of epidemiology at the UK Medicines Control Agency. Our third panelist is Ralph Horowitz, Senior Vice President of Clinical Evaluation Sciences and Senior Advisor to the Chairman of Research and Development at GlaxoSmithKline. And he's also the Harold Hines Jr. Professor Emeritus of Medicine and Epidemiology at Yale University. And finally, we have Alex Walker, a principal of a World Health Information Science Consultants. It's a U.S.-based firm that conducts research in the safety of drugs, devices, vaccines, and medical procedures. He's a former professor and chair of the Department of Epidemiology at the Harvard School of Public Health. So I just kind of wanted to quickly remind you all what topics have sort of bounced around this morning and try to see each of these distinguished panelists will have different takes on these things, different main interests, and besides which we've asked them to just limit their talks discussion to about 5 minutes. So clearly they're going to be making priorities on all the zillions of things we've heard today. I mean, first we heard about the effort that these OMOP researchers have done to create, even to draw up these 399 kind of gold standard across 4 health outcomes. That itself, I think, is a substantial, if they just stopped there, I think it would have been quite a contribution. But no, they devised an OMOP data model. And this OMOP data model is reasonably complex now and complete and has been published and has enabled the transformation of many databases to common analyses. Then they drew up this OSIM2 simulator, the more fancy simulator than they had in previous years. Then they focused on several EPI study designs, and we saw decent examples of the new user cohort and the replication you could do in the literature, various case control options, and the self-controlled case studies as the main ideas. Then we went out with this questionnaire to find out how the experts really do it and whether, and this thing so-called Edin. And we found there's a great diversity among the experts as to which methodologies they actually would choose in any given drug event combination study. Then we learned all about ROC curves and were able to use these 399 results to actually get an idea of the accuracy of the different methodologies. Then that suggested tailoring the analysis to the outcome, then to focus on larger, more complete data examples, tailor the analysis to the data source. And finally, Patrick went over all the sources of data heterogeneity that have bedeviled epi studies from time immemorial. The databases, the methods, the HOI definition, the signal sizes, the parameters. The question then is, what is our research agenda, and what is the reaction that these experts have to all of these items? So let's start out with Dr. Spielberg.
Sure. Well, I should say at the outset what I'm going to say are my views, not necessarily those of the FDA. I should also say, by way of biases, that I've been involved with OMOP since its inception, because prior to coming to FDA, I served on the FNIH board and had the privilege of watching this program evolve over time. My other bias is that I come at adverse drug reactions as a clinician, as a molecular biologist interested in pathogenesis and mechanisms, and although I'm married to an epidemiologist, I aren't one. I'm actually quite overwhelmed by the progress that's occurred with OMOP. We're dealing with incredible complex human biology and medicine. And in the face of that, there is no substitute for having good data. And experimental approaches where you can, in fact, manipulate parameters and examine the effect of those parameters on your outcomes. And I think we've seen that in spades today. As a medical school dean, I'm acutely aware that of the 130 medical schools in the country, we all call every disease differently, that we view patients differently, we consider things serious or not serious differently, our electronic records are all different, and our electronic records are frequently wrong, as are our billing codes. So that we are working in a very complex system. And it's even more remarkable to me just how much useful information has been generated in the course of the OMOP experiment. So seeing database variability, we'd expect it. Seeing parameter variability, we'd expect it. I would like, though, just to put in one additional pitch for biologic variability. And I would refer you to the National Academy of Sciences document in November on precision medicine. I think probably the best evocation of the issues of human variability, both from a genomic and an environmental, a disease, and certainly as a pediatrician from an ontogenetic point of view. So we have an experimental system now to look at the macroepidemiologic issues of ascertainment and hopefully prediction, to which I think we now have the opportunity to integrate into the epidemiology more information on human variability from a genomic, a biomarker, a clinical, a definitional point of view. And having seen so much progress made by OMOP to date, I'm not worried about adding on more data. They can analyze just about anything. You know, I don't know what comes after terabytes, but whatever it is, that's what we're going to be dealing with. But the future clearly is going to require an integrative approach to the biology, the medical care aspects, as well as the outcomes. And I'm really glad to see looking at European databases, because looking across healthcare systems is going to be incredibly informative in understanding disease process, how we define diseases, etc. The other take home that to me is probably the most important is that of trade-offs. We saw that depending on how you set your dials, you can get a large number of false positives, false negatives. What trade-offs mean, though, and I think from a societal point of view we have to realize, is that the different people view trade-offs differently. If you're a patient, you view trade-offs differently in terms of false positives and false negatives. And if you have cancer, you view it differently than if you have a cold. We recently had a symposium at FDA inviting the public in to talk about benefit-risk. And it was a very rich discussion indeed, and one that shows the complexity of how our patients those we serve, and all of us do, how they view the issues of benefit, risk, of ascertainment, of confidence in what we say is safe, what we say is effective. To me as a physician, obviously, the drugs I write for and the therapeutic choices I make and the prescriptions that I hand out are going to be determined very much by information, but I have to understand the nature of the trade-offs. We get reduced to drug X causes liver damage. Well, is it in 10%? Is it in 1 in 10,000? Is it 1 in a million versus the benefits? Hard trade-offs again. And certainly as a regulator, with respect to approval of compounds and certainly of maintenance compounds on the market, we need to think about these trade-offs. Bottom line to me is my hat's off to the OMOP folks who have done a spectacular job in moving us down the pike into more and more complexity, but with that more and more understanding. And I would just add on, the more biology we put into that understanding, the better.
Okay, thank you. And we will have a chance for you to ask questions of the panelists, but let's go on now to Dr. Evans.
Thank you very much, Bill. I'm not an expert. I don't have a PhD. I'm not doctored, but don't let it worry you. Thanks very much for the privilege of being here. What a fantastic job has been done by the OMOP team. This combination of creativity and extremely hard work is one of the most impressive bits of work I've seen in 40 years since leaving CERN in Geneva. Statisticians have made notable contributions to assessments of efficacy using randomized trials since the days of Bradford Hill. Perhaps because it's so difficult, they have, with some exceptions, to be mentioned a little later, tended to avoid studying harms. And it's absolutely great to see good brains and resources devoted to a very challenging area. I've said it before, I think this is as intellectually difficult as particle physics. The work by OMOP has crucial lessons for pharmacoepidemiology in general, and this afternoon will make that even clearer than we've had in the last few minutes from Patrick. So what are the lessons for detecting effects, perhaps not just adverse ones, in large databases by scanning them? What are our lessons? Politicians and some others think it isn't that difficult. You've got all this data and you're going to get more, it's going to be easy. Some epidemiologists think it's impossible. They have vested interests as academics in making sure that you can't do it. You've got to do everything my way. Okay, the not invented here complex. And we had the same when we looked at databases of spontaneous reports. And Bill DuMouchel, with encouragement particularly from Bob O'Neill, as key people with strong statistical backgrounds, making a notable contribution to studying harms, or should I use the euphemism, safety. OMOP shows it's possible, but we need to tread cautiously. It's likely there's no simple answers. H.L. Mencken said, "To every difficult and complicated problem, there is a simple, easily understood answer that is wrong." But I think we've heard today what I would like to think, in some senses, is the beginning of evidence-based epidemiology. That is, that it isn't that epidemiology hasn't provided evidence. We are getting to the point of evidence-based epidemiology in terms of deciding on methods. And I think that's fantastic. We've looked at these areas under the curves, and we know that the cut-off choices, as Dr. Spielberg has said, leads to false positives and false negatives, and we can't ever win. And we also have seen that self-controlled type methods do best. And I would suggest that there are 3 reasons for that. The first, everyone seems to know that they deal with unmeasured fixed confounders, and so it means that the model for analysis is relatively simple. A second one, I think, that has some relation to some of the other things is that missing data on the fixed covariates are then made of minimal importance. The third one is that the errors in the data or the effect of outliers and really influential observations are minimized. And most epidemiologists neglect the effect of influential observations when they do multivariable regression. They have no idea where their influential observations are. And they very rarely, we don't have good methods for detecting them, and they don't do it. And so we've known for a long time that, or for some time, that vaccine effects have been best studied using self-control type methods. And single exposure and rapid onset makes it easy, but they're not the answer to everything. And there are papers by Paddy Farrington just recently in Vaccine that will show you where he thinks, and I think he tends to know a great deal about this, where they can be used for good measurements and where less so. I think in some senses at the School of Hygiene we've led the way in using self-controlled case series methods themselves in studying drugs. And at first we were told we couldn't do that. But we have gone on doing that. And I think they have gains. Now, I think that in terms of one of the things that Patrick said is that the databases tend to be relatively consistent across the different problems. And I think that's partly contingent on the common data model. And some of the consistency there is actually forced by that data model. So where do we go next? I think, as he said, we need to understand where the methods perform poorly. Tukey said, forget the fit, look at the residuals. All right, look where we're getting it wrong. And we need to look at the database characteristics that are dealing with that, the outcome characteristics, and obviously with things like the EU databases with potentially richer information that perhaps have extra stuff beyond that, what effect will that have on the methods? Use of the GPRD in the UK may be very interesting because that is being extended to be the CPRD and increasing in size and complexity. I think there is too much concentration here on optimal methods, and again Patrick talked about the idea of effectively looking at the variance of the area under the curve, and he didn't present any metrics on that but just drew graphs. I think that is important, and not just looking at the highest. I think it's vital for screening that we need to say, let's not look at the optimal method, but the one with the least variance. Positive predictive values are going to be more important than sensitivity and specificity, which relates to sample size and prevalence. I think, again, in regard to what Dr. Spielberg said, we should be measuring absolute effects. Can we actually do some analyses on absolute effects rather than just relative risks? Epidemiologists always measure things with relative risk because they assume they're constant. But for screening, maybe we need to look at something different. That's public health importance. Point estimates have been emphasized. Should we be looking at the lower 90% CI, rather like EB05? Am I out of time? I think we should look at empirical estimates of variability due to the source of data and the method and so on. Can we get an empirical estimate of that rather than just say it? Sander Greenland and others have said that there are lots of other sources of variability, and we need to try and have not just the idea that they may exist, but measure them. And so, finally, it shows us where real sensitivity analysis should be planned in doing a study in epidemiology, and not simply saying it made no difference. You should actually look at the areas where it might make real differences, and where I think we need to have sensitivity analysis within any screening system. We need to actually vary at least one thing and say that my screening method needs to take that into account.
Thank you. So next we'll have Dr. Ralph Horowitz.
So thank you. Unlike Dr. Spielberg, I have not been following OMOP closely since its inception. Up until 18 months ago, I was chair of medicine at Stanford, and OMOP was not really on my radar screen. It was also not appearing, its results were not appearing in the pages of the New England Journal of Medicine or the Journal of the American Medical Association, or for that matter in Lancet or BMJ. And if there are editors of those journals in the room today, and I wish that Jeff Drazen or Howie Bachner were here, I would urge them to pay more attention to what is going on in this experiment. So what would I say about OMOP as a result of what I've learned over the last 6 months or so? Well, first of all, although it didn't get much attention today, I think the common data model deserves a lot of kudos, and the effort to create an infrastructure that supports the kind of analysis that has been carried out thus far, and to applaud the extraordinary transparency of the OMOP team that has put virtually everything from the design of their methods to the code that they use on the web to allow others to follow and to replicate the work that they are doing. From an overarching perspective, I'm impressed that they have created an interesting tension, an interesting conflict between what Dr. Evans referred to as evidence-based epidemiology, or I might prefer empirical-based epidemiology design, with more conventional expert-based epidemiology design. And I think that contrast is at the heart of what will need to be a very intense discussion in our community about how to either reconcile or integrate these different approaches. In the spirit that no good deed goes unpunished, I should note that the OMOP team very wisely, one might say, picked as the basis for their experiment positive and negative controls for which there was little dispute that they could identify in the literature. But if I might butcher the lyrics from a popular song called New York, New York, that center of the universe, I might say that if you can't make it here, you can't make it anywhere. Because the 53 or so topics that form the basis of the experiment really represent extreme examples of drug outcome relationships, those in which there was no conflicting evidence for the positive or negative controls. Most of our examples, of course, represent relationships where that is not the situation, but where there is lots of evidence contributing to both sides of the debate, how well these methods perform, not at the extremes of the distribution where there's no conflicting evidence, but rather in those circumstances where evidence falls on either side of the relationship remains, I think, a fundamental issue that needs to be understood better. In some respects, I would say that the amount of consistency that was observed and which was highlighted by our optimistic colleagues is a source of great encouragement. But there still remains very considerable inconsistency in the findings, depending on not just the issues that were identified by our colleagues here, but by the point that Dr. Spielberg made very strongly, which I hope will get much greater attention, and that is biological and clinical heterogeneity that may contribute very considerably to the variation in results that we've observed. I don't want to go into specific examples, but I do want to take off on the point that was made about Tukey, John Tukey, and refer to a different paper that he wrote a number of years ago on clinical trial research. It appeared in the journal Science. It was a very famous paper on multiplicity in clinical trials. And what Tukey said about that, and I quote, he said, "It is a difficult task to drive the nearly incompatible two-horse team." He was referring now about individual clinical decision-making. And he said, on the one hand, clinicians making decisions have knowledge of a most carefully evaluated kind from rigorous scientific evidence. And on the other hand, they have informed professional opinion, where impressions are gained from statistically inadequate numbers of cases often, and so far as we can see, often should control the treatment choice for an individual patient. The same physician, he said, must be concerned with both what is his knowledge gleaned from rigorous studies and what is his informed professional opinion, often gleaned from treating a single patient. And Tukey said, "I wish I understood better how to help the physician in this essentially ambivalent task." I think that we may be creating a somewhat false dichotomy here between empirically based research design and expert or informed opinion-based research design. Ultimately, we're going to have to find how to reconcile these two. At the fundamental level, it is not sufficient to document, describe, and quantify conflicts. At a fundamental scientific level, we need to understand the source of that conflict. And the next phase of OMOP, I hope, will give considerable attention, not simply to describing the inconsistencies, but to reconciling them, to understand the source and basis of that inconsistency, and help us to make our colleagues in research design better informed experts who utilize both the rigorous evidence that comes from the empirical base and the informed judgment that comes from their own long experience. So thank you very much.
Thank you for very thought-provoking final comment. Alec, what about you?
Thank you. I'll echo many of the things that my predecessors up here have said. I'm going to give a little bit of overall comment. I want to talk about the role of specificity in this exercise, about heterogeneity, and then I'd like to close with a brief word in memoriam following on the death of the propensity-matched new user cohort design. This has been an honest effort to explore what's knowable, and it's very uncomfortable for anybody who's been in the field for a while, and particularly if you've done anything, you've got some territory to defend. And what the OMOP team has been doing has been really taking a flamethrower to the whole edifice of observational epidemiology applied to drug safety. I think we have to, you can think about how you're going to react to that, but I think that what we've got here is an opportunity to start substantially rethinking what we're doing, and I think we actually have a mandate to do that. The fact that there's a diversity of expert opinion, which was documented in some of the things that OMOP did and other people have been doing as well, I think is testimony to the fact that it's not obvious what to do. And OMOP has shown us now empirically that many of the things are been ill-based. Okay. This is really an exercise in epistemology. What is knowable? And I think that we have to take a real step back and ask ourselves that and make sure that we're not going off half-cocked. Some specific areas on specificity. I know that we're in early days in the OMOP experiments, but I'd say that if you were actually running a signal detection system, a specificity, and you're looking at all possible outcomes, or even a handful of drugs, a specificity of less than 99% is just completely unacceptable. You don't have the personnel to work up 1% false positive rate if you're looking at all possible outcomes. Even if you have 100 drugs and 100 outcomes, that's what, 10,000 combinations, you can calculate out what it takes. And so that I think the starting point if we're talking about a real implementation, is what we can afford, and what we can afford is not is how much time, staff, resources do we have to deal with false positives, and so that's where I think we have to start. And that leads you to now, from what we've seen, what's a reasonable expectation of sensitivity for this. Now, back when we started talking about these things, I don't think anybody would have said there would have been 95% sensitivity. You're not going to get almost all adverse reactions. Probably wouldn't have even said 80%, like most of them. Half of them, well, some of us might have thought that maybe half of the clinically important events we could identify once the system was really optimal and really going well. I think the fact is that we're going to be down around 5 or 10%. That in order to have acceptable specificity, that we're going to be dealing with sensitivities on that order, and we do have to think about whether the whole enterprise is worth doing at that level as opposed to investing resources in better spontaneous reports. I mean, I think we really need to think about that. The next point I'd like to address is heterogeneity. I think that the heterogeneity between data sources and methods and so forth is a wonderful opportunity to investigate what's going on. I think that with the very large data resources that are being used, that we can get away from positing probability models and start looking at what are effectively random effects models to quantify in some way the degree to which we understand things, looking for heterogeneity within strata and between data sources, particularly but within strata that ought to be giving the same result given our prior expectation, may lead us down to finding some large effects in small populations that are masquerading as small effects overall. I think there's a lot of hope there, and I think that subgroup analysis combined with science may be very hopeful for that. Finally, on propensity-matched new user cohort designs, at least as a method for screening for effects using outpatient insurance data, that fell by the wayside, and I think there was every reason to be optimistic about it, but it's left off, what we kind of knew in our guts in terms of drug epidemiologists. Let me call it anticipatory prescription. That is, doctors make choices for reasons. If you're in your data, it looks like there is some element of random variation left after you've controlled for everything in the doctor's choice, and it just means you don't have enough data. Doctors don't flip coins. And usually they'll think about what's going to happen in making a choice. And I think that there's good reasons, and rather than fighting as to why the OMOP data shouldn't be true, I think that we need to recognize that this may be over and done. Thank you.